So with that, I'd like to introduce the first speaker, who is John Jumper. John Jumper is a senior research scientist at the Google DeepMind team in London, UK, and he was lead of the AlphaFold team on protein structure design. John, the floor is all yours.
Thank you. And you should be able to see my slides now. And we'll be talking about AlphaFold really in two contexts. One, I think, is an example of how our scientific knowledge can really inform how we build machine learning systems and how do we bring the important experimental and theoretical work of science back into our machine learning. And then I think also as an example of how it's being used and the impact it's having on the community and maybe how we might think about these types of problems going forward. And so I'll go ahead and get started. And AlphaFold, that you've probably heard of, is in a sense a simple system. And if you think of it like a mathematician, you might say it's a really, really simple system. It's something that takes what is very accessible on the left, protein sequences, and through a big fat complex arrow produces on the right predictions of protein structure, sometimes and often highly accurate predictions, on which you can build your experiments and base some of your understanding of the system. And this is really nice, it's really clean as a problem, and it's also really, really important. But what goes in the middle is really challenging. And we've known for a long time, and we've known since Anfinsin, that such a thing should exist, that the sequence should determine the structure, of course, up to exceptions. But how exactly we do this and how we handle the complexity of biology and of experimental conditions and everything else in getting between the left and right-hand sides has been a grand challenge for a long time. And so, of course, if we're talking about machine learning, we should start by talking about data, and we should talk about it in two contexts. And of course, this is the incredible work and the incredible foresight of the Protein Data Bank, which is now about 50 years old, recently celebrated its 50th anniversary. And as you probably know, protein structures are extraordinarily difficult to determine experimentally. We know less than 200,000 deposited in the main database, but very excitingly and very importantly, that this is an incredibly diverse set, that the Protein Data Bank represents the output of the experimental community, thanks to the effort of the community to really say that to publish a structure, you must deposit it here. And so it contains this incredible diversity and is also the basis of being able to do evaluation. That when you talk about machine learning, and really, really good machine learning must come with really, really great evaluation and understanding of it. And this was enabled by the Protein Data Bank and by the CASP community to do blind assessments to really understand what works and what doesn't work. And this is really the key ingredient of even having a machine learning problem in the first place. You must define what you want to produce and how you're going to figure out if you've produced it. Now with that, I can show you a picture of the AlphaFold high-level diagram, and it's already extremely complex. And if you're not in the field, you shouldn't think of it this way. You shouldn't think about all these individual blocks and connections and their meaning exactly. You should really kind of step back and think about some of the principles of how we want to understand structure. And so the first principle, and this is certainly older than the AlphaFold work and a lot of great work over many labs over the years, is, well, let's not think about the Anfinsin problem exactly. Let's not talk about how the sequence determines the structure of the energy landscape via really physical things. Let's also say, well, the structure has a very strong influence on the evolution of the protein. Structure is conserved over long time scales. And so there's been the field of evolutionary structure prediction, including coevolution, where we try and understand the structure from its effect on evolution and try and find the structure based on saying here are many, many homologs that share strong structural similarity, and this is a much more rich and powerful information source. And that's one strand that came in, but we've had these types of ideas for some time, and they've made a lot of progress, but there was still quite a bit missing. And part of what was missing is ability to handle the complexity. And there's been this enormous story that surely you haven't missed of machine learning. That's probably why you're here. And one way that I like to think about machine learning is it's given us these incredible gadgets and blocks and pieces of understanding data, of making really, really complex functions. And, you know, attention is the most common now. There are many others. And these blocks are incredibly powerful. They work in speech and language and understanding cat videos. they're so generic that they know nothing about proteins, that they'll require the data to tell them everything about proteins, but we only have about 200,000 protein structures. So in a sense, we want a place to insert our kind of understanding and creativity into this. And so really, as we built AlphaFold 2, what we were really thinking about, and this is a huge departure from AlphaFold 1, where we had a very off-the-shelf machine learning system together with a complex system that tried to insert the science around it. We tried to put our understanding of physical insights or geometric insights into the machine learning. And as an example of how we did this, and this is one of the main blocks in the system, really said, okay, well, how should you think about residue pairs, the kind of pairwise interactions or core to physics? How should we think about evolution and multiple sequence alignments as how we start as our starting place for understanding evolution? And then how do we kind of make those pieces of data interact in appropriate ways? And here we're taking the gadgets like attention and adapting them to say, okay, well, if you're doing, for example, attention, if you're trying to find relationships in the MSA, well, what you currently understand about the pairwise structure, what are contacts, et cetera, should really inform that. And so when we process the MSA, we modulate that processing by our understanding of the structure. Now, having developed possibly an updated understanding of the MSA, of the multi-sequence alignment, the evolution, well, we can now try and insert that information in the structure in this alternating way. And what we do is we build this basic block that we call evoformer that has these kind of semantic operations between evolution and geometry or physics and alternates them. We don't really say what the network should do, but we build, because it can learn that from the data, it will learn that in the attempt to produce PDB structures. But what we are doing is really saying this is the ways you should communicate. It's kind of like being a good manager. You don't always tell people what to do. Sometimes you simply tell them that they should go talk to someone else. And so when we built this, and I won't go into the details since this is a short talk, but as we did more and more things to align how we built our neural networks with what we understood about the protein problem, we saw dramatically increased efficiency and accuracy in predicting the answer. And in a sense, if we had a PDB that was 100 times as big, I'm quite sure we wouldn't have to do any of this, that it would learn structure pretty directly. But at the size of experimental data we have, really have to build some of our understanding and cleverness into the machine learning system. And this large trade-off between using lots of data when you have it or using lots of your own human intelligence when you have less data. And it's a continuing trade-off within the machine learning field. Now, we built a system, it predicts structures. I haven't gone into many details, but the most important thing I've left off is, as you know, it doesn't always give the right answer. There may not always even be one answer. How do we understand the confidence? And especially as we're saying that experimental biologists should go do a different experiment because the AlphaFold prediction explains some fact about their protein. What is that reliable? And in what cases are that reliable? And what about the prediction is reliable? And so we put a lot of effort into building two different types of confidence measures. And you shouldn't think of an AlphaFold prediction as strictly right or strictly wrong. Like all models, it kind of induces some hypotheses about the world. And sometimes the model is saying, I believe these are trustworthy hypotheses, and sometimes I believe they're not. And so like the simplest and most direct, and you'll almost always see predicted structures colored according to some measure of confidence. We use PLDDT because LDDT is a well-established measure of local structural quality developed externally. So PLDDT is the prediction of LDDT. And what we find is this is a really, really convenient way to get a kind of top-level understanding of these are trustworthy or untrustworthy areas. So blue, the dark blue is the kind of confidences where we expect even the side chains to often be correct. The light blue is we expect the cartoon to look right and the side chains to look wrong. Yellow is a really unfortunate thing where we probably aren't right, we're not sure. And then interestingly, the lowest confidence of this measure is quite commonly associated with disorder. And we see, and low confidence and especially some smooth versions of it are extremely strong predictors of disorder that are almost certainly more accurate than IUPRED and competitive with special purpose disorder predictors. And so what we're really saying is, and it's kind of obvious when you think about it, that the disordered regions must be low confidence because the network hasn't found anything because there's nothing to find in terms of structure. But it is really important to emphasize, and this is part of like correctness isn't just one thing. An individual atom is not correct. It's correct in relation to others. That we have an example on the right, which is a lot of light blue, right? We think the backbone is correct, but you can kind of tell if you're familiar with any of these class of proteins, that doesn't look like a very plausible overall domain packing for this. And so even though all of those domains are thought to be correct, it's almost certainly not the correct relationship of them, and very likely this particular protein is flexible. And these kind of considerations led us to develop a second confidence measure that I think is much more powerful. And I should say, if you're using AlphaFold structures and interpreting them, simply never interpret them without looking at the PAE, or predicted aligned error. And here, this is a pairwise measure that is roughly, for a given ij pixel, is residue j in a correct relationship to residue I. And what's really, really interesting about this is within a domain, normally this will be dark, meaning high confidence. Everyone will be confident relative to each other. But for interdomain interactions, you see quite a bit of difference. And in the example like we had on the previous slide, you can see that almost none of the domains are thought to be in correct alignment relative to each other, which is true. This is certainly not a correct domain packing, and there may not exist a domain packing. But you can often find interesting and predicted interactions. And so there's actually been some really great work, I should have updated the slides for it, from the Marcotte lab, showing, for example, that the PAE confidence is an extremely good predictor of chemical cross-linking concordance. So high PAE indicates that there'll be a high percentage of, say, cross-linking violations, and low PAE has almost no chemical cross-linking violations. So this is a really important measure in how you think about this. And in all these cases, when you're doing machine learning, the model is making a prediction. If you're lucky, or if it's designed well, you'll have an idea of confidence of that prediction. But it's also kind of incumbent on you of, even within that prediction, when you're predicting really complex objects, like a protein structure, that different relationships will be confident or non-confident. And so it's something that needs to be handled with care as we build these into larger systems or build biological understanding. Now, the other kind of interesting question is, well, what about the fact that Anfinsin is an ill-defined problem? What is the structure of the sequence? Isn't exactly correct. We don't know. What about post-translational modifications, apo versus holo, oligomer state, everything else? And it's worth saying AlphaFold does not produce apo structures. That's a misunderstanding of the task it was given. The task it was given, it said, here is a sequence. The sequence has been deposited within some PDB structure. It could be an enormous cryo-EM structure in which this is a small percentage of the overall structure. This could be an NMR structure. This could be many, many other things. And AlphaFold is always trying to predict how it would appear in PDB. So if this is a heme binding protein, even though AlphaFold can't produce hemes, wasn't told there was a heme, it will leave heme-shaped holes, or very similarly for metal binding sites. Even interestingly for homomers, it often does a very good job. So you see the prediction in the middle, the T1080, the CASP target ID. AlphaFold, actually, we predicted one of those chains, and it was able to produce the intertwined structure that when aligned, when three copies are aligned, you can see it's clearly an intertwined trimer. And so we found actually AlphaFold is highly accurate at producing homomer structures even in isolation. Now there's work to understand, because it doesn't tell you if they are homomers. This doesn't work as well, for an example, like on the right, where heterotypic interactions stabilize the green chain. And here we can't really predict that. Although I will say, and I won't talk about it as much in this talk for time, we've developed a system, AlphaFold Multimer, that you can also freely use that predicts protein-protein interactions often quite accurately. And so if you have a system that is really stabilized by heterotypic contacts, you really, really need to tell it the heterotypic partners and use the Multimer system to build it. But all of this is kind of saying, be really, really careful when you think about these machine learning systems. Think about what they're trying to do in the sense of how were they trained, and then also think about how you're using or understanding them. Don't, things missing aren't necessarily not part of the prediction in a certain sense. In some ways, they are inferred. And it's complex, and it's still, I think, an evolving area of how we understand these. But it has been used really effectively. A couple pieces of work from external labs, both understanding the context in which, for example, post-translational modifications occur, and then also some interesting work that says, well, actually, if AlphaFold leaves, say, a glycan-shaped hole in a protein in a known glycosylation site, then we can just build that in. And then there's been external work, AlphaFill and others, on how do we build, how do we kind of complete these structures or add the implied context back into these structures. And this is something where I think it will evolve rapidly over the next few years in terms of our ability to predict these directly as opposed to working afterwards. But all of this can be done. I think there's going to be a lot of really great work in how do we understand cellular context from the predicted protein structures. There's also been, I think, some of the most exciting work using AlphaFold has been in structure determination itself. So within the crystallography community, it is very, very heavily used for phasing. And then within the cryo-EM community, AlphaFold has kind of an NMR-like behavior where it's often locally correct, or it's finding local interactions and the global structure arises from local interactions, as opposed to crystallography or cryo-EM, where the local structure is extracted by the superposition of many particles. So blurry or lower resolution, for example, cryo-EM, or cryo-ET, like the enormous nuclear pore complex, really benefits because it both validates AlphaFold models and then AlphaFold is providing atomic detail. And so there's been some really exciting work in integrative structure determination in bringing together this kind of the resolution revolution, especially the mid-range of that, together with atomic details from predicted structures. And so this is a rapidly evolving area. And I think kind of stepping back, like this is the AlphaFold story, but I hope that in 10 years, AlphaFold will be seen as kind of the moment that opened the floodgates of machine learning, really changing how we think about structural biology and larger biological questions. And I like to think of structure prediction and machine learning in general as an amplifier for the incredible work of the experimental community. And so it's an opportunity to take kind of sparse experimental data and complete the picture. And so I think what will be really exciting in the coming years is how do we start designing experiments in order to train machine learning models? We're already seeing that with multiplexed assays. And then how do we really start to think about this interchange between experiment and machine learning? And then how do we use the machine learning and when is it reliable? And I think this will drive a lot of exciting progress because biology is enormous and enormously complex. And I think we finally have the kind of mathematical and computational tools to meet that complexity head on, but it's still a lot of difficult and exciting work to come. And with that, I want to thank the many, many people that made AlphaFold possible, incredible team at DeepMind that worked on both the methods and our release of the human proteome and the larger AlphaFold database, which I actually didn't talk about today, but I assume many, many of you have used. And want to very much call out our collaboration with Imble to make these predictions available. And they've been incredible partners in how we build this, how we explain these results to experimentalists, and how do we make sure that we're really being responsible. We feel, this enormous weight of we want to save experimentalists time and not lead them down blind alleys. And I think it's been really, really exciting to see what people have built on top of that work. And with that, I want to say thank you to everyone and especially the organizers.